AP Bio, and welcome to a video overview of Chapter 8, Photosynthesis, the process that feeds the biosphere. And when I say that, I mean the process that allows all heterotrophs, including us, to have chemical energy to undergo metabolic processes. So photosynthesis is just simply the process where we take solar radiation coming from the sun and convert that into chemical energy, knowing full well that A, matter can't be destroyed nor created, the same with energy, and also that energy tends to go from high to low in terms of usable forms because heat is lost. Directly or indirectly, photosynthesis is going to be the process that drives the rest of the biosphere in terms of our ability to do work. Now, autotrophs like plants and algae are organisms that sustain themselves without eating other organisms, and this is because they're able to use organic molecules such as CO2 to undergo processes that create types of macromolecules such as glucose that fuel cellular respiration. And most plants are photoautotrophs, meaning they use the energy of the sun to make those organic molecules. Photo meaning light, auto meaning self. And heterotrophs, meaning other energy, obtain their organic matter from other organisms. So these are the primary consumers up to tertiary and quaternary consumers that obtain the chemical energy from plants. And they also are the ones that require oxygen as that oxidative agent to break down glucose in cells. Now, in terms of a little bit of anatomy, we're going to be looking at plant cells, primarily in leaves, which have a high surface area to volume ratio to absorb lots of sunlight. And we're going to be looking in chloroplast cells. And within these mesophyll cells, there are an organelle called chloroplasts, which occur in a double membrane, just like mitochondria. And we have stacks of thylakoids, which are an organelle that we call granum. And these are going to be where we have our light harvesting complexes that absorb light to produce chemical energy carriers that will allow us to fix carbon dioxide. The stroma is the fluid that surrounds these thylakoids. So they're almost cells within cells, giving us good evidence that these were once independently alive prokaryotic organisms that probably formed some association with another cell. So in a nutshell, photosynthesis can be reduced down to carbon dioxide and water in vapor or water in the atmosphere coming together with light energy to form glucose, oxygen, and water vapor as well. Chloroplasts are going to be splitting the H2O that generally is formed during cellular respiration into hydrogen and oxygen, and they're going to use those electrons from hydrogen to make sugar molecules. So in a way, we are sort of doing the complete opposite of cellular respiration. Rather than oxygen absorbing electrons, we are going to be taking electrons away from oxygen. And we can sort of see how the reactants in cellular respiration are low energy, very stable molecules. Carbon dioxide and H2O are very small, and we're going to be forming very large, unstable, but high energy products such as glucose. And there's two parts to photosynthesis. There's the light reaction and the Calvin cycle. The light reaction is going to be the light part where we harvest light, and this is analogous to the ETS in cellular respiration. And the Calvin cycle, the synthesis part, is where we produce glucose. In many ways, this mirrors the Krebs cycle in cellular respiration. In the light reaction, we're going to be splitting water and releasing oxygen, and we are going to be reducing NADP plus to NADPH. So again, this is analogous to NADH in cellular respiration. And we are going to be generating ATP from ADP by adding a phosphate group called photophosphorylation. This is going to be acting very similarly to the electron transport system in cellular respiration. The main idea behind this slide is to make sure that you do not get confused between the two, because there are many similarities. in a nutshell, light is going to enter mesophyll cells in the leaves, enter the thylakoids that exist within chloroplasts. Water will be inputted into the light reaction. We will remove electrons from H2O, forming oxygen. And again, this needs energy because H2O is a polar molecule. O2 is a nonpolar molecule. So it's very hard to take electrons away from that very stable low energy molecule. As a result of that energy transformation, we are able to reduce ATP and NADPH, which enter the Calvin cycle that take carbon dioxide and pump out a CH2O molecule, a precursor to glucose. The ADP and the NAD+ are recycled back into the light reaction. So let's talk about light. The light that is going to be used as energy for this process fits within the electromagnetic spectrum, which we know as radiation, and primarily we're going to be talking about visible light, which has wavelengths that produce the colors that we see, and generally light can be understood as discrete particles of photons with very specific energy levels depending on the wavelength of that light. What do I mean by that? When we look at visible light, we have colors ranging from violet to red with wavelengths of about 380 nanometers to 750 nanometers. And this really can be used to extrapolate the frequency at which light hits a particular surface. A shorter wavelength with a higher energy is going to be hitting an object more frequently, and a longer wavelength with lower energy hits an object less frequently. Plants need to absorb this light and use it to drive photosynthesis, and they've created, through evolution and natural selection, a very complex system that allows them to harvest this light to create chemical energy. And when we see light entering mesophyll cells into the chloroplasts, reflected light is light that is not going to be absorbed by these photosystem complexes. Generally, green light is the energy source of visible electromagnetic radiation that is not utilized by plants, while other colors such as red and blues and purples tend to be more utilized. We call the light that is used absorbed, and the transmitted light is the reflected light. And why is this? Well, plants have evolved a series of proteins that they have embedded in membranes that are really good at absorbing different frequencies of light. There are a couple different types of pigments on here. Pigments are another word for proteins in the photosystem complex. We have chlorophyll A, chlorophyll B, and then things like carotenoids. I always remember carotenoids because it sounds like carrot. And what you can see on this visible spectrum is the amount of light that each of these pigments absorbs relative to the wavelength of light. So you will notice that chlorophyll A and chlorophyll B are very good at absorbing high energy radiation from 400 to 500 nanometers, and they're moderately successful at absorbing 600 to 700 nanometers of light, which are the lower energy intensities. Carotenoids, on the other hand, are a little bit better at absorbing that moderate amount of energy. You will notice that there is a very large gap within this area here where the 500 to 600 nanometers of light aren't really absorbed. And this is because photosynthetic bacteria that existed before plants evolved pigments that absorbed this moderate wavelength of light. So as plants evolved to undergo photosynthesis, they obtained pigments that allowed them to outcompete that bacteria for other wavelengths of light. And this is used to create what's known as an absorption spectra. Basically, when we look at a plant and a leaf cell, what is the general summary of the light that they can utilize for photosynthetic activity based on the concentration of each of these carotenoids or pigments and what they do in terms of absorption. So within the photosystem, we have a series of complex events that are basically going to be taking electrons and exciting them, and that excitement is going to be used to generate not only heat, but also a series of redox reactions to power ATP production and NADPH production. As the energy of the electron moves down, so too will the ability to use it to do chemical work. Now, within the photosystem complex, embedded in a phospholipid membrane are lots of different pigments. And I like to think of these pigments as a series of slightly more electronegative and slightly less energetic proteins that are going to be able to absorb different frequencies of light. And once that light has been absorbed, it's going to pass from a high to low, high to low, high to low, high to low protein. Each time that energy is going to be reduced just a little bit. And when I say reduced, I mean the intensity is going to be lowered. So light hitting and being absorbed by pigments high up in the photosystem complex probably are going to be light photons that are in the high energy wavelength. And then light that we absorb lower down in the photosystem complexes are going to be light photons that tend to be lower energy. And this is because we want to reduce the chance of heat and also energy combustion within the cells. Plants can actually undergo heat stress if they're not able to absorb light efficiently and without huge gaps in the energy of those electrons. Now there's two main photosystems that exist within most chlorophyll cells. There's photosystem 2, and photosystem 2 has a special pair of protons that make it really good at absorbing 680 nanometers of light. And I apologize, I said protons, I meant proteins. And this is called PS2 or P680. That means that it is going to be able to use energy really well in the spectrum, but notice most energy that plants absorb tend to be a lot higher in terms of energy wavelength. And then there's also photosystem 1, which is really good at absorbing energy at 700 nanometers. And this is called PS1 or photosystem 1 or P700. And it's really good at using energy in this part of the visible spectrum. Again, we have all of this energy absorbed here as well. So we need to use protein complexes to reduce that energy in small steps. A large jump from energy here to here would release a tremendous amount of heat. So the linear electron flow, which is very similar to the ETS and cellular respiration, can be broken down into a series of steps. One, a photon hits a pigment or a protein, and its energy is passed amongst pigments, going from high to low until it excites P680. That P680, when it is excited, is going to donate an electron to the primary electron acceptor, which exists in the photosystems. And because those electrons have been removed, P680 now becomes an oxidizer that helps split H2O with some enzymes, and those electrons are then passed on to the P680, hence reducing it to P680 without the plus. And oxygen is formed as a byproduct. So we can see that in the system we have light hitting a pigment. The pigment passes the flow of energy from high to low. So this might be 350 to 400 to 450 to 500 to 600. And finally, we're able to pass it along to P680. And P680 will use that energy to excite electrons, at which point it needs to be reduced. And it's going to use enzymes to help split water and pass electrons back into it because it's now in a more electronegative state. Once it has those electrons, the process can happen again. The electrons that we excited with light energy then flow through a protein complex within the membrane system, going from high to low energy, and we use that change in energy from high to low to pump out hydrogen ions, forming ATP, much like the electron transport system in cellular respiration. And then we get to Photosystem One, where the same thing occurs, and light passes on from high to low, so 350 to 450, 500, 600, 650, 700. I'm just making these numbers up, but that's how light is being broken down in terms of high to low energy. And then the same thing happens again where we excite electrons, and we have the second system, the electron transport chain, reduce electrons in terms of taking their energy from high to low, and that's going to help us make some NADPH. Now, chloroplasts and mitochondria both generate ATP by chemiosmosis, but there are different sources of energy. This is really important for understanding the differences between Chapter 7 and 8. Mitochondria obviously transfer chemical energy from food, breaking down glucose in glycolysis, pyruvate oxidation, and the Krebs cycle, to then power a series of protein complexes to make ATP. Chloroplast transforms light energy into chemical energy for ATP. There is differences in spatial organization of where we put those hydrogen ions between mitochondria and chloroplasts, but they are both using the ATP synthase protein pump. In mitochondria, protons are pumped into the intermembrane space. Remember, there's a two-membrane fold in mitochondria, and that drives ATP as they diffuse back into the inner part of the mitochondria. In chloroplasts, protons are pumped into the thylakoid space and drive ATP synthase as they diffuse back into the stroma. So what we're going to be doing is we're going to be pumping hydrogen ions into the thylakoids, and then they're going to be pumped out back into the stroma, fueling that ATP production. There might be another image showing that. Excellent. So hydrogen flow out, ATP produced via chemiosmosis. And here's what the system really looks like in a nutshell. We're going to use Photosystem 2 to split water and Photosystem 1 to absorb electrons from Photosystem 2. Those processes are either going to make hydrogen ions be pumped against their concentration gradient into the thylakoid space and back out through ATP synthase, or we're going to be reducing those electrons to create NADPH. Now, the Calvin cycle is going to be taking those two chemical molecules, ATP and NADPH, and they're going to be creating the building blocks of glucose. So we are going to be creating an anabolic cycle, not a catabolic cycle. And the sugar that it builds from these molecules will be the precursors to glucose. We're going to be using energy and reduction of those molecules by these two chemical agents. Now, carbon enters the cycle as CO2, and it's going to be leaving as G3P, which is a precursor to glucose. It generally takes 3 cycles or 3 molecules of CO2 to make 1 molecule of G3P. Phase 1 of the Calvin cycle is carbon fixation, and that's where we are going to be incorporating CO2 into Rubisco biphosphate using the enzyme Rubisco. So we are going to be taking a 1, 2, 3, 4, 5 carbon molecule, adding CO2 to Rubisco, and we are then going to be making a 6 carbon molecule. We are then going to be adding some ATP energy and some NADPH energy to that molecule, which is going to be reducing it, adding energy. And at that point, the molecule that was 6 carbons is going to be split into 2 3-carbon molecules, and G3P is going to be split out. And then we're going to be taking another G3P molecule, and we're going to be regenerating that initial Rubisco biphosphate to add complexity to it. I think that, like the citric acid cycle, knowing things like G3P or biphosphate glycerate or any of the enzymes, maybe Rubisco is important, but these steps aren't as important as understanding that we use ATP and NADPH to either reduce or regenerate carbon molecules to take CO2 and pump out G3P. So there are some evolutionary adaptations, much like anaerobic and aerobic respiration, in different metabolic processes that we see in cellular respiration. And the primary evolutionary adaptation of plants is to adapt to dehydration or temperature. And adaptations to dehydration is a problem for land plants that often have trade-offs, especially with photosynthesis. On hot days, plants have to be really concerned about evapotranspiration, meaning water loss through their stomata. Remember that water is a polar molecule, which means water lost in the leaves are going to pull on water in the shoots and in the roots due to adhesion and cohesion. But when we close the stomata, we limit photosynthesis because no new CO2 can enter the mesophyll cells. And that causes oxygen to build up in the leaf cells. This then causes cells to transform in plant cells to a process called photorespiration, which is quite wasteful in terms of the storage of energy, and it can be damaging to plant cells. So most plants that exist on the planet are what we call C3. They have the initial fixation of CO2, and they form a 3-carbon molecule called G3P. In photorespiration, the Rubisco adds oxygen instead of carbon dioxide in the Calvin cycle, and that produces a 2-carbon molecule rather than a 3-carbon molecule. And again, this is happening in C3 plants when it's too hot or it's too dry out. And photorespiration decreases the photosynthetic output. It consumes ATP, it consumes oxygen, and it releases CO2 without providing any new sugar to the plant. So in a way, photorespiration is plants' version of cellular respiration, where they're consuming oxygen and releasing CO2, and they're not making any sugar molecules. So we have one evolutionary adaptation called C4, where we minimize the cost of photorespiration by incorporating CO2 into a 4-carbon compound. An enzyme in the mesophyll cells has a really high affinity for CO2, and it can fix carbon even when CO2 concentrations are very low. And these 4-carbon compounds are exported into bundle sheath cells, where they release CO2 that is used in the Calvin cycle. So we can have C4 plants taking the carbon dioxide, and they fixate that CO2 in a bundle sheath cell, where we then undergo the Calvin cycle. So we're spatially separating the fixation of carbon and the reduction of carbon to make sure that we efficiently use all of the energy. We're sort of splitting the two processes in 2 different cells to make sure that all carbon dioxide is used. And again, we're able to do this when the stomata are closed in plants under heat stress, because this cell, we are using an enzyme that is very, very, very attracted and has a very high affinity to CO2. Alternatively, we can have CAM plants where they fixate the carbon dioxide at night into a precursor molecule, and then they pump that take that organic acid, and they then create CO2 from it during the day to produce sugar. And this is more of a temporal separation. So I hope that addressed some pressing concerns or questions about photosynthesis. In a nutshell, I feel like you should be able to explain this chart quite well in terms of reduction and regeneration. I hope that you're able to understand this complex in terms of a series of energy transformations and reductions. And driving this, again, is going to be this notion of different proteins in the photosystem that absorb different amounts of light.